Log on, tune in, find out. Another good idea from Cambridge.
Thank you very much for the introduction. So today I will be talking about variational methods in image processing, integro-differential models, and applications to histology and MR imaging. So the talk is basically divided into two parts. So the first part is basically problems in image processing, BBL2 based integro-differential equations, a few theoretical results about BBL2.
A little louder, Prashant.
Huh?
A little louder. No, that won't help.
Oh, okay. So BBL1 based and BBL2 based integro-differential equations, and a few theoretical results and modifications to these models. Okay. So just a quick thing here about images. So digital images would be basically treated here as two-dimensional analog signals, and today we will talk about only black and white images, which is basically a function from R2 to R. And when I talk about intensity, f of x would be the intensity level at that point x, which varies from 0 to 255, which basically depends on the number of bits that you are using. So here images will be postulated as L2 images. Here we see a typical image of Lena, and this is the Lena seen as a graph of a function. So classically we have these few problems, image denoising, where you have an f image which is given to you and you want to find out what is the noise in it. You want to find out the true image, eliminate the noise. So f is equal to u plus eta, eta is the noise, we need to get back the denoised image. So f may be blurry and noisy, so that means you have this blurring parameter k, blurring operator, which could be a convolution with some kernel, and then you have image segmentation problem, identifying the components in the given image, so which is related to the H detection. Multi-scale image representation, which is what we are going to talk about here, is basically you have an image and you want to extract different scales in this image. That means, for example, here is the city of Mumbai, and here is the same city which is seen at some height, and then you go down, you see more details, and you go again down, here you have more and more details showing up. So I would call this thing as a large scale image, a little bit more finer scales are here, and this is finer scale. So just to point out, clarify the terminology. So this is a multi-scale representation of the city of Bombay, for example. So multi-scale a representation would be basically given or the image f, you want to produce a family of images ut with some scaling parameter. Okay. So there are many approaches to solve these denoising, deblurring, etc. problems. So two methods, of course there are many, but today we will talk about variational approaches, Tikhonov regularization, greedy algorithms, etc. PDE based approaches, diffusion, Perona-Malik, and so on. So these approaches are of course related, and here is the famous Rudin-Osher-Fatemi decomposition, where f is given to you, f is a given image, and you want to basically denoise. So the original intention was to denoise this image, get an image u lambda, and v lambda is the supposedly the noise, but of course a denoising can be thought about as decomposition, right. So you have f and then you have u and v, which is basically a decomposition of f, and lambda is a scaling parameter. So here is the problem that we are going to solve, decomposition of f into u and v, where you want to minimize this BV norm, and f minus u square integral is the fidelity term. So here lambda acts as an inverse scale of the u lambda part. That means smaller the lambda, the scales in u lambda are larger, and so on, and vice versa. So, here U is basically contains the smooth part of the image, prominent edges, and V contains the texture, finer details, etc. So, there are many variational other variational problems methods that we will not get into. So, heat equation, basically everybody likes heat equation. So, here you have this image F and you run the heat equation, you get something like this. The problem with heat equation is that it denoises, but of course destroys all the edges, right. So, the edges are gone, but the noise is also gone, right. So, the problem here is we cannot distinguish, it cannot distinguish between noise and boundaries of the region. You do not know where to stop, because it will keep on blurring the image. The reason that it does that, it is an isotropic diffusion, we lose information about the edges, right. So, Perron and Malik proposed this anisotropic diffusion method, where you have they introduced this function g, which is basically diffusion controlling function. The idea here is to preserve the edges. That means, if you have smooth regions, that means the gradient is weak, you need isotropic diffusion, and if you have prominent edges, then the gradient is large, we need to control the diffusion. And the example of a function that does that is something like this, e to the power minus any function that starts with 1 and goes to 0 with time. Okay. Of course, it is not well posed. A simple modification by convolution with g is it makes it well posed, and this is a modification due to Quetelet, Morel and Cole, 1992. So, here another idea here is you have this Laplacian, it can be decomposed into tangential and normal components, right. So, you have this tangential component here. So, Alvarez, Lyons, Morel's model uses this idea. Here the idea is basically you have this image, you do not want to diffuse in this direction, because that will destroy the edges, and you want to diffuse only in the direction which is tangential to this edge. So, this is exactly what they do, and they also use this the same function edge control, the diffusion controlling function to improve this thing, right. So, and the results, as you can see, is fantastic. So, again the problem is we do not know where to stop. A solution was proposed by Nordstrom, electrical engineer from Berkeley. So, he introduces this f minus u in this Perron and Malik and proves that it has a steady state non-trivial solution. So, Rudy Nausher-Fatemi, going back to that, is you solve this equation, and the time dependent Euler-Lagrange differential equation is this, and you compare that with Nordstrom modification of Perron and Malik, they look almost similar. In fact, if you use g is equal to 1 over 2 lambda s, it is exactly the Euler-Lagrange differential equation of ROF. So far we have discussed so many models. So, we propose this novel integro-differential model. Here we have integral of u x s is equal to f plus 1 over 2 lambda t times the curvature term. So, this is an integro-differential equation as opposed to PDEs. The motivation, so here also the scaling function here is, it is basically a function which is increasing function, monotone increasing at our disposal with some conditions. Okay. So, the motivation is you have this IDE and I want to get away from variational formulations. Questions is what is the motivation behind this thing, and how do we start this, how do we stop, and what does this scaling function lambda t mean. So, hopefully we will answer these questions. The motivation basically comes from Tadmor, Nazar and Weiser scheme. So, here is again the Rudy Nausher-Fatemi decomposition with a intensity quanta. So, this is basically a constant, and you try to solve this problem with this quanta here. So, and then you decompose f into lambda tau times u lambda 0 plus v lambda 0, and then again you take this v lambda 0 and decompose it into again different parts using lambda 1 now, and you keep on doing this. So, basically you produce a multiscale representation, and after n plus 1 steps you get something that looks like this. It is a nonlinear multiscale decomposition with this lambda. So, again going back to this Rudin-O'Shea-Protemes, kth step in TNV reads this, right? And here V lambda k is this function here, this entity, and TNV iteration is written as this. The telescopic sum of the bow gives us this equation in summation form. So, V lambda n is here. So, you put this thing here and you get a nonlinear equation that looks like this. This motivates us to write the following model, okay, and we will see what this lambda t is again. Just to a quick note as to how to solve this thing is basically we denote this script u as the integral of little u, and this is the script u that is something that we want. This is the image which would be denoised image, or the image that gives you the scale of the function. Okay, so basically you solve it by putting it into a summation form and iteratively solving it using this fixed point iteration. So, these are the results. So, this is the original integro-differential equation. So, note here that in this, when time is t is small, you get only the large scale image, and as t increases, more and more details come into this integral of u. So, here more and more details are appearing into this sequence. So, a few theoretical results before we modify this model. So, the star norm is a dual of the BB norm with respect to the LP inner L2 inner product. So, using this norm we can prove this theorem. So, for the given IDE we have this integral of u as this the script u, and capital V is basically f minus the script u. Then the size of the residual, the star norm is dictated by this 1 over 2 lambda t, which is basically thought, you can think about this thing as the measure of how much noise it has. Theorem number 2 is about the energy decomposition. So, for the given IDE we have this for L2 image f, we have this energy decomposition, that means a weighted BV norm integral plus the energy of the noise is exactly equal to the energy of the given function square. Okay, and the next one is about the convergence. So, basically here you have this energy decomposition. What happens if t goes to infinity? If we prove that the V L2 norm of V goes to 0, we get this convergence of this integral, and which is this theorem, and this is the condition that we have to apply on this increasing function. So, it has to be sufficiently increasing function, sufficiently rapidly increasing function. Basically any exponentially increasing function qualifies to be this function. Okay. So, an open problem here is this is proven for BV functions. What happens if f is in L2 is not known. Modifications to BV L2. Suppose, okay, going back to the heat equation, Perron-Malik is this modification of, well, it is not Perron-Malik, Catullion's, but anyway. And then what happens if we apply this g function outside this divergence term? And well, basically it is the same thing, except that we have this lambda t as a different function which depends on the position. This is the result of this modified filter IDE model. You have this, again the large scale images are there, but the edges are more pronounced, right? And again is the same behavior, more and more details come into this sequence as t increases. Okay, now again MR images. So, here is the brain of a person. So, the original form is like this, again this is a large scale image, but the edges are gone. But if we apply this edge enhancing function here, we retain the edges even at small t. Another modification is the tangential smoothing modification, where you have this heat equation, and then we know that Laplacian can be decomposed into tangential and normal components. We again use the tangential component with this thing. Can we do the same thing with this IDE? Of course we can, we will try. And this is the tangential component, and maybe we can apply this g function also. The results are like this. So, this is the original version. As you can see, this is the f given f, and the noise is reduced, but ultimately it comes back. Of course, that is the behavior of this IDE. But if we apply this tangential smoothing, we suppress the noise and we retain this edge. If we apply this filtering, we enhance the edge even better, but ultimately the noise will come later. So, deblurring with IDE, which is something that I don't want to spend too much time about, is basically the same thing, except now you have a convolution or a blurring operator k. So, the TNV schemes with scheme with deblurring is something like this, which gives rise to this integro-differential equation with this k star. The result is basically this: you have this blurred image. You go from here to here by applying this method. The problem here is you need to know what k is, otherwise it will not work. So, the details of this thing is published in IPI, multi-scale image representation, 2009. So, what happens when we apply this thing? What do we do if we have BBL1 decomposition? Let's try to see this thing. So, BBL1 model, there are so many names attached to this model, of course, and this decomposition is contrast invariant, and the space generated is more geometric in nature, which is the result of Janis and Toglu, 2005, a fantastic paper. Okay. So, this is BBL1 hierarchical decomposition with intensity quanta. Let us do this thing. Here it is a little bit complicated because you have the signum function, right. So, the summation form that we get by putting this thing here is a little bit different, but anyway, so this motivates us to write this idea. Let us look at the result. The results are very similar. There is not much difference because it is the same thing. You have more structure, the large scale structure appearing before the finer scale, that is okay. But it is not quite evident as to how much it is different from the BBL2 unless you look at something like this. So, here again you can see that this is really the scale space. The large scale structure appears before the finer scale, irrespective of the intensity of this tiny structure. So, which is quite interesting, as opposed to compare that with BBL2, where you have this block appearing and the intensity is increasing gradually rather than abruptly. So, which is different. A few theoretical results about BBL1 IDE. So, a similar property, similar theorem here. Here we have the signum of V star, the star norm of the signum of V is 1 over lambda t. Recall that for BBL2 IDE we have this star norm of V itself was 1 over lambda t times m. Okay, so that is a little bit different. Theorem number 2, here we have this energy L1 energy decomposition, which is quite interesting because it is very similar to the BBL2 energy decomposition, except that you have L2 square and you have L1. So, which is nice, I believe. But the theorem number 3, which is showing that this guy goes to 0, is still open. So, if you the things are complicated because of the signum function. So, I do not know. So, modifications again the same similar modification of BBL1 IDE. We have this. So, you have signum function, this BBL1 IDE. We have the filter diffusion or edge enhancing function. If you apply that, you again get the same kind of behavior. You enhance the edges, and BBL1, if you, I do not know if you could see that, it tries to capture the large scale structure here. Compare that with this BBL1 original form. You have the large scale structure, but the edges are gone. Okay. Again, BBL1 with tangential smoothing is even nicer because of the contrast invariant. It does not depend on the, because this is salt and pepper, well, not salt and pepper, but the noise is suppressed irrespective of its intensity in BBL1, which did not happen in BBL2. Okay, and compare these results with this, Again here you have the noise appearing at t is equal to 18, and the edges are not retained properly. So, let us connect the dots. Here we started with heat equation, and then we looked at this Perron-Malik. Then we looked at this modification of Perron-Malik of Nordstrom's PDE, and we saw that it was quite related to this Rudin-Usher-Fatemi model. And then TNV is basically Rudin-Usher-Fatemi model implemented in multiscale hierarchical decomposition, and that led to our novel integro-differential equations and BBL1, BBL2 and its modifications, and combining with these other modifications. Okay. So, this is basically the first part of the talk that we wanted to talk about. So, epilogue is basically advertisement as to what I was doing and what I will be doing. So, my life at UCLA with human placenta. So, placenta is basically an organ which grows along with the baby, where the umbilical cord is connected to the center of the placenta. Okay. So, we will talk about this movie that we produced about this histology of the placental tissue. There are other projects that we did with Pascal and Luminita Vese, which were presented at IFPHLA, blood tissue segmentation. We will not talk about this. The study of vasculature structure. So, basically what the problem here is, you have this set of images which are produced by staining this tissue, placental tissue, by some chemicals, hematoxylin and eosin. What happens here is the blood is written in red, and the nucleus is stained blue, and everything else is kind of in between, pink. Here the problem is, you have hundreds of such slices like this, and registration is a problem, because first off, they are very big images and the blood vessels move. So, you cannot exactly match or register everything. Yeah, thank you. So, that is a problem. Landmark registration would not work, because the images are scaled, sometimes rotated and things like shifted. Sometimes you have torn tissue. So, in this case nothing works, right. So, idea here is to postulate that the images, the set of images, the difference is they are rotated, scaled and translated, right. So, this is the transformation. So, we try to basically look at the following minimization problem. This can be solved. So, here is the reference image, this is the target image. So, you go from S to T, and we want to find out this transformation. This is the scaling, this is the rotation, this is the translation. So, we solve it by Newton's method. This is impractical because the images are too big. So, you approach this thing by multi-level approach. So, you look at a small scale image and then use these parameters to find out, to use it in the next level, and so on. So, we look at the movie that we made. Let us see if it works. Control L. Okay, so here we see this movie where these are the blood vessels which are moving and merging, and the problem here is, first off, having this thing was one project, and another project is, you see these blood vessels are moving and we track them. Okay, so tracking of the blood vessel is another problem. Okay, so here we can have this, we want to have a fully automated algorithm, but of course it is too much to ask for, I couldn't do it. So, another way is to have a skilled person, a doctor, MD, to scan, to trace all these images manually. It is very expensive time wise and money wise, because these people are trained, highly skilled people. Other way is to have a moderately skilled person trace all of them, but this is too much because minimum wage is too high. And yet another way is to have a moderately skilled person trace few of them and use these images to somehow trace in between. So, these images are not high quality images. The tracing is tracing made by a moderately skilled person like me would be something like this. So, you have a blood vessel here, and this person is not doing a very good job, doing something funny here, and there are gaps. But anyway, and the point here is, given this tracing, you want to find out where this blood vessel goes in the next image, and the next image, and the next image. So, the approach is like this. So, you have initial rigid registration like the movie that we made. Then we have this initial inaccurate initial inaccurate tracing, and then we look at the local problem, local rigid registration, and then we use Chan-Weisser segmentation to find out this boundary. And the problem here is you need to find out these parameters properly in order to find out exactly one blood vessel. So you kind of construct some heuristic algorithm so that you find exactly one object in the next image. The result is this. This is the fifth image in the sequence. So it works very well, and it actually worked very well. Even the fact that here you have an image which is torn, right? So the tracing is satisfactory. It is submitted to SPI medical imaging. Let's see what happens. So compare that with this one, which is certainly better. This is the last slide. This is what I am doing right now in University of Toronto, and so this is basically a heart vessel, left ventricle, and the problem here is to find out the scar tissue and the healthy tissue and something in between, so that we can treat this patient well. So this is the work by Detsky, Paul, Alexander Dick, and Graham Wright, and I will be hopefully doing something in that direction. Well, thank you very much. I really appreciate for this this chance that Adrian has given and the organizers have given me. Thank you. Yes.
Can you go back to a slide with several squares there? Oh yeah.
Yeah.
So what kind of original images do you use for this one?
Oh yeah. So this is given to you. You cannot have this one and get that. So it is an algorithm which extracts scales rather than going from this image to this image. You cannot do that. So this is given to you, and you want to find out large scale images, large scale structures in this image. Yeah.
That's it. Thank you very much for that.
Yeah, no, ask me.
In the placenta movie,
Yeah, the movie.
so no, I don't need to re-see
Yeah.
it, but from one frame to the next, what is happening? We're going to a different slice, or it's a different point in time?
So what
or-
happens is this placenta is here, right? And this is the umbilical cord. Umbilical cord is their blood vessels, and this blood vessel goes inside the placenta and forms a tree, right? So inside this thing is like a tree, right? And then the thing that you have seen is the vertical slices of these things, and the area that you have here is 1 inch by 1 inch, and the distance between them is
So we're
is-
progressing
it is progressing from
slice to
here
slice.
to here vertically.
Okay.
So the problem is
Follow the blood
to
vessel.
follow the blood vessel and to see how they move. Yeah.
Well, thank you very much for spending the afternoon. Springtime, and there is an announcement to make.
There is not